So in this lecture, we're going to be going through chapter 17, which is viruses and immunity. In addition to giving you guys a little bit of history, we're going to be talking about what is a virion, how do we know that it's the viruses that are making us sick, or how is it discovered that it wasn't some bacterium or toxin that was making us sick? What are the components of viruses? DNA versus RNA viruses, how do viruses replicate, and how can we treat or prevent viruses? Not necessarily in that order. This isn't a history class, so I try not to show pictures of old white guys, but these guys were pretty important in the discovery about the fact that it is actually viruses that are causing disease sometimes. Certainly still bacteria and parasites can cause disease, but many of these things, even after the discovery of what's called the germ theory of disease, in large part thanks to Mr. Pasteur here, we were unaware that it was viruses that were contributing to what was making us sick. And so what happened was Adolf Meyer here showed that you can take an extract from a plant. So you can take a plant and expose another plant to that first plant's extract. And if that first plant was sick, the second plant is also going to get sick. Then we had Ivanovsky and Bejernik down here, and what those guys showed was if you remove everything from that extract, even things as small as bacteria, it would still make the other plant sick. And so what that showed was, well, it's not the plant material making the other plant sick, it's not any bacteria making the other plant sick. What is in this sick plant that can get other plants to be sick? And this is the discovery of the virus. So Interestingly, if you remember from the discovery of the structure of DNA, Rosalind Franklin is here again because she did some of the original crystallography on viruses, and she suggested that the virus had a hollow component to it. She helped us to figure out what the structure of viruses actually were. So these guys are more like 1800s guys. Rosalind Franklin worked in the 1930s. 1930s, 40s. And her work, again, we didn't discover that she was correct until after her death, unfortunately. Here is the smallpox virus. Here is a smallpox vaccine. Smallpox is one of the few diseases that we've actually managed to get rid of in the world. Smallpox does not exist naturally anymore. They still have it in labs. And the history of vaccination is pretty fascinating. So if we go back a slide, Louis Pasteur here, his initial discovery was that if you took a virus like rabies, which rabies is fatal, by the way. If you don't get vaccinated or if you don't get treated for rabies and the virus gets to your brain, you are going to die. And so you can imagine 150 years ago, that was pretty horrifying. It's this virus. They didn't even really know it was a virus yet, but this thing that if you get bitten by wild animals, you start going crazy and ultimately you die. So what Pasteur did, though, was he took the rabies virus and he passed it through many, many generations of rabbits. And the virus actually adapted itself so that it became a rabbit virus, at least that particular strain. And what that did was it attenuated the virus to make it so that it was not deadly for humans. And then he took some of the serum from one of those rabbits and injected it into a boy that had rabies, a boy that was going to die if they didn't do something. And because of his intervention, by giving the boy the attenuated virus, it helped his adaptive immune system, which we're going to talk about later on, but it helped his immune system actually prepare itself so that it could fight off the full deadly rabies virus that he was also infected with. So again, here is the tobacco mosaic virus that they discovered was causing these plants to actually get sick using those pretty brilliant experiments. And again, these pictures we weren't able to take pictures of viruses with electron microscopy until, gosh, the late 30s. So it was well after we were actually able to visualize these things, or well before we were able to visualize these things, that they figured out, okay, there's something else going on here that's making these plants sick. It's not bacterial, it's not toxins, it's something else. So this gives you a sense of the size of a virus, and so this makes it look like if you just glance at this, you might think, oh, the virus is about the same size as a mitochondria or a bacteria. It is not. This is much, much smaller. This is 100 nanometers compared to 1 micrometer. So a flu virus is actually an order of magnitude smaller than mitochondria and bacteria. These are very, very, very tiny things. So bacteria can be visualized using light microscopy. Viruses, you actually need electron microscopy to be able to see them. They are very, very, very small. Here's another interesting one. This is the Ebola virus being looked at with transmission and scanning. I don't know why I couldn't think of the word scanning. Transmission and scanning electron microscopy. Yeah, here's just a picture of a virus, and you can see it. They've colorized it so it looks very, very, very sinister. But there it is. There is a picture of the Ebola virus. So, looking at COVID-19 with an electron microscope, you can see it's got that characteristic shape that kind of looks like the Canvas logo. Somebody pointed out pretty hilariously. But there are several components to this, okay? It has a thing around its genetic material called a capsid, C-A-P-S-I-D, okay? And it also has this RNA inside of it. Coronavirus is an RNA virus. There are also DNA viruses out there, and we're going to talk about the mechanism or why it is an RNA virus. It has this membrane protein here, and it has these spike proteins here. So the main things you should know are it has a capsid surrounding its genetic material, and it has some way of interacting with the cell that it's going to infect. This figure 17.5, you don't need to worry about all this text, but what this is showing is that viruses can be incredibly complex, like this bacteriophage that looks like a weird alien ship, or they can be relatively simple, like this adenovirus over here. All viruses share a capsid and genetic material. Okay. They also are going to have these glycoproteins that they're going to use to interact with cells. They interact with specific kinds of cells. So a bacteriophage is going to be infecting different cells, different kinds of cells than an HIV virus. So viruses are generally species specific. They can transfer between species pretty often, but they are generally species specific, or like Pasteur showed with the rabbits, they can be sort of bred, if you want to think of it that way, but they can be made to be specific to one line or one kind of species, and they're also generally cell specific. Okay, and again, these are just general rules, but generally it's going to attack only one type of cell from one type of species. So these images, by the way, I will link the article where I found them, but this is a fantastic way of understanding how coronavirus actually infects us. So I'm going to put a link to that with this video. I highly recommend giving it a read. It's very, very simplified, and it is just phenomenal. Okay, so here's a host cell here, and here's the coronavirus. The coronavirus is going to interact with the cell, and it's going to wind up getting its genetic information into it, and it's going to make many, many hundreds of, if not thousands of copies of itself. This is not to scale, by the way. Coronaviruses are actually going to be much, much, much smaller than a eukaryotic cell, but just for the sake of looking at these things. So the first thing that the coronavirus needs to do is find this thing here. This is called an ACE2 receptor. That's just what this particular virus interacts with. And so when it finds this ACE2 receptor, it's going to bind to the cell. It's going to bind to the cell, and it's going to inject or allow its RNA to be released into the cell. Okay, if it doesn't find an ACE2 receptor, it's going to move on. It's just going to kind of keep on floating throughout your body. It's not going to actually interact with it. But here we have it. Here is a coronavirus interacting with an ACE2 receptor. So once it gets inside, it brings its own RNA polymerase with it, but it's going to use the cell's machinery to make copies of its genetic material. Okay? So copies of specific RNA sections that tell the cell how to make more viral proteins. These proteins were going to be used to build the next generation of coronavirus. So, by the way, some people have heard of this drug, Rimdenzavir. The Remdesivir actually targets RNA polymerase, and that makes it much more difficult for the RNA to be copied inside of the host cell. And so, if you were curious about what some of these drugs do, that's what one of them does. So, here we are at the endoplasmic reticulum. So, we are inside of the cell now. Here's the nucleus. And these ribosomes are making viral proteins from that RNA that the virus injected. So the virus put in the RNA, it put in its RNA polymerase, made a whole bunch of copy of this RNA, and that RNA is going to go where RNA goes, to the endoplasmic reticulum, and now your cell is making protein. Your cell is making the virus. That sounds kind of spooky. Maybe it should. A lot of viruses work this way, though. Actually, all viruses hijack your cell's machinery so that they can make copies of themselves. Okay, so we're up here on the top of the endoplasmic reticulum. We're just going to pan down now to the bottom. It doesn't really matter. This is kind of happening throughout, but she did a really good job of separating things up in these images. And so you have all of these viral proteins that have been made by your cell. And what's going to happen is you're going to wind up with basically entire RNA proteins, or not RNA, you're going to wind up with entire viruses inside of your endoplasmic reticulum. So here you have a complete virus, and that virus is going to be loaded onto this thing, which maybe you remember from before is called a vesicle. And those vesicles are going to be taken and dropped off at the outside of the cell, where those viruses are going to go infect other cells. They're going to go find more ACE2 receptors. They're going to be volatilized and coughed out or spoken out by people, and they're going to go find more stuff to infect. One virus infected cell can produce hundreds to thousands of new viruses. Here's just another picture of sort of how that works. You have your virion, which I should have mentioned before. Virion is an infectious viral particle. So whenever you see little pictures of viruses, those are virions. All right, so if somebody says virion, that is what they are referring to. They're just referring to one of these particles. Okay, so this thing is going to get into the cell, and then it is going to add its RNA to your nucleus. Okay, that RNA is going to get copied a whole bunch of times. Okay, and then it is going to get translated. So it's kind of like transcription, but really just copying your RNA. Remember from before, transcription was taking DNA and making an RNA copy of it. Really here we're just making copies of that RNA from RNA. So it's not quite transcription, but it's kind of like transcription. But then the cell is going to undergo translation. It's going to make all of those viral particles. Okay, it's going to make all of those viral particles, which are going to be taken and put into a vesicle and then ultimately released from the cell. Okay, and again, these proportions are super, super wrong. These virions are much, 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 much smaller than the cells themselves. This is just kind of the textbook's way of saying the same thing, but we'll go through it really quick. This is in the case of influenza. This is no longer coronavirus, although flus can be a type of coronavirus. So the influenza virus is going to bind to those receptors, and actually it's going to be brought inside of a vesicle. Okay. The The contents of the virus are going to be released, including viral RNA, which is going to enter the nucleus, and it's going to be replicated by a viral RNA polymerase, which we've kind of already gone through. The viral mRNA is going to be used to make viral proteins, which are going to be assembled into a new virus, which is going to be released from the cell. So let's see, did we cover everything? So what is a virion? It is a viral particle. It is an infectious particle. It's the little thing that binds to your cell and makes you sick. How do we know that it's viruses making us sick? Well, we have pictures of them for one thing, but those initial experiments really told us that it wasn't bacteria that was actually making us sick. It was something else going on with the plants that were making them sick. Virus components, they have capsid, they have glycoprotein that they're going to use to interact with receptors on cells, and they have some sort of genetic material. RNA versus DNA viruses, I guess I didn't actually cover this, but viruses can have either RNA, which is going to get turned into protein, or they can have DNA, which is going to get transcribed into mRNA and then translated into protein later on. How do viruses replicate? I went over that several times. That's really the only one I'm going to ask you about, although there are others. That's the one the coronaviruses use, so I figured that's the one we would be most interested in. How can we prevent or treat diseases? Well, the answer to that is by utilizing vaccines. And we're going to get into why vaccines actually work later. But the basics of that are they just tell your body, hey, here's the kind of invaders that are coming. Here's what they look like. Here's how you can protect yourself against them. So it's kind of like a wanted poster for your immune system. Okay, that's the end of this lecture. Yeah, I will see you guys in the next one.